Population genetics. Population genetics. Scientists building on the research of Darwin and Mendel came up with a whole new branch of biology called population genetics. Population genetics examines how the frequencies of alleles in populations change over time. The inheritance patterns and principles found by Mendel explain how variation occurred in populations. Natural selection explained why some alleles became more frequent in a population than others. Genetic variation. Populations having genetic variation can evolve by natural selection. Genetic variation allows populations to adapt to ever-changing environments. Populations with no genetic variation face extinction, ultimately, because they are all alike. If an environmental change cannot be handled by a population with no variation, all will die. If the population has genetic variation, there could be organisms that will survive, and the population will continue on. The Hardy-Weinberg Equilibrium theory. Two scientists independently worked out a way to measure evolution in populations. G.H. Hardy and Wilhelm Weinberg approached a biologist and said, "Hey, you need some more mathematics in your science." So they came up with the Hardy-Weinberg Equilibrium theory that mathematically measures gene frequencies in populations from generation to generation. Allele frequencies. Hardy-Weinberg equilibrium measures microevolution rather than large-scale macroevolution. Microevolution is the change within small groups of organisms or species over a short period of time. Macroevolution is the evolution of large groups of organisms over a long period of time. By measuring allele frequencies, which are how often dominant and recessive alleles show up in populations, from generation to generation, the Hardy-Weinberg equation can be used to determine if a population is currently evolving. Micro means small, and macro means large. Hardy-Weinberg equation. Hardy-Weinberg equilibrium is calculated using the following equation. P squared plus 2pq plus q squared is equal to 1, where p squared is equal to the frequency of the homozygous dominant genotype, p is equal to the frequency of the dominant allele, 2pq, the frequency of the heterozygous genotype, q squared, the frequency of the homozygous recessive genotype, q is the frequency of the recessive allele. Calculating the equation is saved for AP Biology. Population according to Hardy-Weinberg. The Hardy-Weinberg equation can determine the following about a population: phenotypic frequencies in a population, genotypic frequencies in a population, allelic frequencies in a population, how many individuals exhibit the different phenotypes and genotypes, and if a population is evolving. Conditions for Hardy-Weinberg. The Hardy-Weinberg equilibrium equation can only be used with the following conditions to determine if the population is evolving. Populations have to be extremely large, allele populations are stable, there are no mutations, mating is random, and there is no natural selection. Used for comparison. The conditions to use the equation are very restrictive and are only possible in rare conditions. Example to follow. Most real-life populations are not found to be in Hardy-Weinberg equilibrium because they are always evolving. The reason for using Hardy-Weinberg is to set up conditions for fictional populations. By doing this, we can use it as a comparison to see the reasons for evolution in real populations. Exception to the rule. A small number of real-life populations are close to Hardy-Weinberg equilibrium. They are either not evolving or only have slight microevolution. The Galapagos blue-footed booby is one of those populations because the alleles remain stable in the population. Populations like this are helpful to scientists that study them as a comparison to populations that are evolving.